Good afternoon, everyone, and welcome to today's webinar on how to make the most of your PrimePCR with your real-time thermal cycler, hosted by Lasec in collaboration with Bio-Rad. We are pleased to have Luke Forbes with us today as our speaker. Luke is the Distinguished Field Application Specialist from Bio-Rad with a strong background in genomics as well as digital PCR. Before I hand over to Luke, just kindly note that the chat function has been disabled. However, I would like to ask you or to encourage you to participate by using the Q&A features to ask questions. And these will be answered at the end of the presentation. So without further ado, please join me in welcoming Luke. Over to you, Luke.
Thanks, Michelle, and thank you everybody for joining. I realized after we created this webinar that it's April 1st. It's not an April Fool's webinar, it's a real webinar for real people. So yeah, that's thanks for the introduction, Michelle. As Michelle said, my name is Luke. I'm Field Application Specialist at Bio-Rad. I think some of you would have known me before. And I support the genomics, so anything to do with PCR, whether it's extraction all the way to PCR, and then also cell biology, flow cytometry as well. Okay, so today we're specifically going to be discussing PrimePCR, which is a product from Bio-Rad that's sort of new to the South African market in a sense. So we want to start by sort of gauging your familiarity with the product or just general pre-designed primers and probes and what you kind of make use of in your lab. So to start, just with an introductory poll, I'm going to launch the poll. Or Michelle, I'll do that. And you'll see on your screen this first question. Who has performed qPCR experiments in their lab before? So who of you that's in attendance so far, who has performed a real-time PCR experiment in their lab before? I'll just give you guys a couple seconds to answer that. Just helps me to gauge what sort of discussion we're having today. Then the second question, which I'm going to work through, of those of you who have, how many of you have designed or used your own primers then rather than designing your own? So if you just scroll through the poll, you'll see a second question. You can select, I design my own, I use pre-designed, or I use both. So you design some of your own and you do pre-designed. And then the third question, how many of you have specifically used Bio-Rad's PrimePCR assays before? I have, I haven't, but I need more info. I haven't, but I have the intention of using them. You can select option. And then what's your biggest challenge when running real-time PCR experiments? Is it primer design, reproducibility, data analysis, or something else? This just also helps me to kind of get an idea of what in future, what kind of webinars we can look at. We have options for gene expression, sort of troubleshooting, what to look out for, choosing reference genes. We can also sort of discuss those technologies in a bit more detail or issues you might be having. So you can just type in for the fourth question, what is your biggest challenge? You can just type in whatever it might be, whether it's interpreting data, the wet lab part, reproducibility, etc. Or is it designing primers? You can let us know. Okay, that's the poll. I hope all of you have had a chance to just select sort of an option within the poll. And then I can see there's so far quite a few responses. So I can see use both. Okay. Okay, great. So just to go through some of the answers, who has performed real-time experiments in their lab? It's just above 60%. So 67, 64% of you have attended, are running real-time experiments in your labs. And of those that are running real-time PCR experiments, we're looking at around 67% that use both. So Most prefer using pre-designed, and others use both options as pre-designed and they design them themselves. They are most of you are interested in Bio-Rad PrimePCR assays, but you want more information on them and you have the intention of using them. And then if we look through the biggest challenges, we're looking at sort of mostly primer design, reproducibility being a big issue, quality issues, and reproducibility of results. Okay, so I'm going to leave this poll active while we run through today's sort of session. And then at the end of the session, if you have any specific questions, either with regards to the poll or additional questions, obviously we'll get to them as well. Okay. So for the background, what sort of components are required for a real-time PCR experiment? Okay, you need your templates, which is a nucleic acid. This can be DNA or RNA. Then you need your primers, which are complementary to this sort of specific amplicon of interest, and that's where primer design comes in, which is what we'll sort of be discussing today, and how PrimePCR can help you there. Then there's probes, which you have a hydrolysis probe, which is also specific to your target sequence. If you're using TaqMan or probe-based chemistry, it's an additional design that you have to do for a real-time PCR experiment, and it's obviously complementary to your amplicon of interest. Then you've got your DNTPs, which is added in complementary fashion to your DNA, double-stranded DNA that's split into single-stranded DNA. And then finally your enzyme, which is Taq polymerase. And your enzyme's obviously got things like buffer to create an ideal sort of working environment for the enzyme. So we're not going to be looking at the template, and we're not going to be looking at DNTPs, enzyme, or buffers, but specifically we're going to be looking at primers and probes. Obviously that's where PrimePCR falls in the workflow. And if you look at sort of a background of a real-time PCR workflow, you've got to define your target and your reference gene. You plan appropriate controls, maybe something like a no template control, positive control. You make sure you know which detection chemistry you're going to be using, whether it's cyber-green or probe-based. Then when it comes to sample prep, you've got to extract high-quality nucleic acids. You assess your quality or quantity with sort of wavelength or fluorometry. Then you can use maybe a DNA treatment if you're using RNA samples and you want to remove DNA. If you're using RNA, you'll do reverse transcription, where you convert your RNA to cDNA using an appropriate reverse transcription enzyme. And it's important to also remember your priming strategy when you're selecting a reverse transcription enzyme. So is it random hexamers? Is it oligo-DT? Is it a combination? And obviously each of those has their benefits, which we can discuss on a separate webinar. Then when it comes to real-time PCR setup, you've got your traditional approach, in which case you need to design and validate your primers, or you've got your PrimePCR advantage, which comes in at skipping validation and using pre-optimized assays, which obviously take a lot of time savings into effect, and then preparing your master mixes, running your thermal cycling and optimized protocol on a calibrated instrument, collecting your data at the specific phase, and performing a melt curve analysis. And of course, you only perform melt curves if you're running a cyber-green based experiment, because you're trying to confirm that it's non-specific, that it's a specific product and there's no non-specific binding. And then finally is your data analysis, which you're setting thresholds, calculating efficiency, performing relative or absolute quantification, and applying some sort of normalization strategy. Now, if we look at specifically PrimePCR, we're going to look at the PrimePCR fundamentals. What is PrimePCR? What is the product? How can it fit in your workflow? What kind of solution does it offer you? So when we talk about PrimePCR, it's gene-specific primers, and they're in different product formats. So there's the individual assays format, which is a single target for specific gene of interest. And there's obviously cyber-green based assays, and there's probe-based assays as well. There's pre-designed for humans, mouse, and rat. Those are the pre-designed assays we have. And there's options to choose an intron spanning or exon specific designs depending on what you're looking to target. Then you've got your DNA templates, which is at the top right there, which are gene specific synthetic matched DNA templates designed to be used as a positive real-time PCR control. So matched meaning that the primer that you would buy, purchase through PrimePCR would have a synthetic matched DNA, meaning that the same primers would target that specific positive control, and you can then obviously determine if your experiment is working correctly in those kind of settings. We also have pre-amp assays that provide sort of unbiased pre-amplification of target genes, and this is for when you are faced with small quantities of cDNA. And for every single PrimePCR assay that we have, we do have a corresponding preamp assay that's available. So these assays are just basically provided at a much higher stock solution. And they basically target the genes that you're looking at, they pool them, and then they preamp to create more quantity for a precious sample. Then in the bottom left, we have experimental controls. Now, under the experimental controls, we have a full complement of controls, whether it's synthetic DNA for positive controls. We also have things like DNA contamination control assays that are species-specific controls that target a non-transcribed region of the genome, and they're used to determine if you maybe have genomic DNA contamination. You've got positive PCR controls that target a DNA template to determine if your sample contains an inhibitor or other factors that's maybe stopping the PCR reaction occurring. So you can also get RNA quality assays, which is a pair of assays that target sort of on the same transcript at different locations with different amplicon sizes. And then the two assays are used as a pair together, and the difference in the CQ value can indicate whether your RNA is being degraded or not, and it may be negatively affecting your expression results. And there's also reverse transcription control assays, which introduce a synthetic RNA template into your cDNA synthesis reaction to evaluate your reverse transcription performance and confirm that that's happening fully. And then, of course, there's reference gene assays, which can be used to normalize for input variations in your RNA, especially important if you're doing gene expression as well. Then after the individual sort of assays, which are single tubes, we have pre-designed plates. These are really helpful for customers. There are about 7,000 different plates to choose from that we offer. Note that these plates are just pre-plated cyber primers, so they're lyophilized in each well. So you can just add your cDNA, your supermix, and you can almost instantly be doing real-time PCR. The pre-designed plates are obviously in those same three genomes, so house, human, mouse, and rat rather, and they can be purchased in 96 and 384 format. And then what is nice about these plates is there's no minimum order quantity required, and they're available for pretty much any real-time PCR instruments except 48 well instruments, because obviously we only offer a 96 well, or something maybe like a QIAGEN rotogene, which is obviously not thermal block based, so also wouldn't fit. Now, it's quite interesting to note that the Bio-Rad PrimePCR portfolio is the largest pre-designed panel portfolio in the industry, and it allows sort of customers to begin a brand new screening project. They can use a plate directly related to their specific research, and you can look for target genes without having to scour multiple literature resources, databases, and all these kind of things. You can just look at a single plate sort of in your research focus, and you can narrow down a specific subset of genes, and then you can evaluate them on that plate. Or if you want to do downstream, you can look at individual assays, or maybe using a custom plate. And just to note, we also have, they're called the SAB, or the sort of QIAGEN replica plates, which are PrimePCR pre-designed plates using the same gene target lists as the SAB arrays, but with a much better performance and much higher validation. And they've just been taken by looking at the top ranked genes within a disease or a process, and then the reference panels as well. Yeah, so just to note that we do have reference panels specifically for validating or checking reference genes if you're running a qPCR gene expression experiment as well. And then we've got the custom PCR plates, which are personalized plate layouts, and you can select the targets yourself. They're available also in the 96 and 384 format, and you can choose from a selection of validated assays. Why do we use CyberGreen as opposed to Pro? Because it's much easier to transport, it's much cheaper ultimately for you to run, and it's cheaper to buy a plate with single assays in each well. And then if you want to do multiplexing or something in the long term, then moving over to a pro-based experiment once you know what genes you want to target. Now, this is some of the plates that we offer within our pathway and disease panels. So there's thousands of unique pre-designed panels. There's 4 main categories that they sort of fall under: diseases, processes, metabolism, and protein function. Diseases specifically like cancer and mental disorders, major clinical research priorities. Then there's the processes like fundamental cell mechanisms, apoptosis, transcription, development. Under metabolism, we have complete metabolic pathway coverage like lipids, carbohydrates, and amino acids. And then under protein function, signaling proteins and functional categories like cytokines and kinases. So the benefits in terms of running these on real-time PCRs, you've got no need to manually select genes for pathway studies. These are expertly curated gene sets, meaning that your pathway is fully covered, has full coverage. And then there's some panels across multiple species if you're looking to do translational research. And again, they're compatible with multiple instruments. So what are the key features and benefits of PrimePCR? Well, what is the cost of an unvalidated assay to you? Time is probably the biggest one. Researchers spend weeks optimizing and troubleshooting poorly designed primers. There's also a resource drain because you're making use of costly reagents and precious sample that has been reused during your experiments. Publication delays, also giving you unreliable data that requires experiments to be repeated, extending research timelines, and data integrity issues. Questionable results can sort of undermine your research projects ultimately. So what we offer with PrimePCR is we have a rigorous testing sort of process, and this eliminates the typical 3 to 4 week primer design and validation cycle. It saves around 15 to 20 hours of lab work, which is normally carried out per target gene. It reduces the risk of experiment failure due to primer design issues, and it follows the MARCI guidelines, which we'll discuss a little bit further, which helps you to meet publication requirements right from the start. We also guarantee specificity on our products, which are preventing sort of false positives or off-target amplification, which is really crucial for gene families with sort of high homology across them. And it obviously reduces cross-reactivity with pseudogenes and things like that, which is critical for accurate expression and biomarker discovery. We also ensure that we have optimal efficiency. So we have a 90% plus efficiency range, which ensures that you have accurate quantification across your concentration ranges. It's essential for your sort of fold changes, calculations, etc. And it enables you to be sure that you are confident that your targets are being amplified within various different species as well. When it comes to sensitivity, we detect down to 10 copies. So we really go for low abundance transcripts, which is for things like biomarker research, and early detection is really important, and enables you to sort of work with limited clinical samples that obviously you can't get more of, as well as analysis of rare cell populations. And then a really great feature is we do have fully documented validation data. So we provide all the evidence for regulatory submissions in your publications. It enhances the reproducibility between labs and studies. It simplifies your troubleshooting when an unexpected error might occur, and helps you to meet requirements for journals in terms of assay validation documentation. If we look at the second big sort of differentiator with PrimePCR, we can look at the sort of how it saves your time in a qPCR workflow. So I think the overall time reduction is between 40 to 60% faster. You're eliminating around 5 steps, which are obviously included in the standard workflow. And what would normally take you around 3 to 4 weeks can be done around 3 to 4 days with PrimePCR. You eliminate the design phase, which is 2 to 3 days of bioinformatics work. You can remove the validation phase, which is 1 to 2 weeks of lab work. You remove the troubleshooting loops, which sort of no feedback loop. You keep having to go back and forth. Now, if you look at this in the real world, in step 5 on the standard qPCR workflow, you'll see there that designing primers normally takes you 1 or 2 days. Ordering primers, another 2 or 3 days, at least probably 2 to 3 weeks of wait time. Then you've got to do your primer evaluation, which is another 3 to 5 days. And then does it work at step 8, 1 to 2 days analysis, and then a redesign cycle if not working. Whereas compared to looking at PrimePCR at step 3, you just use the PrimePCR lookup tool, which is 15 to 30 minutes. Step 4, you order your PrimePCR samples, which is typically around 3 to 4 weeks, and you proceed directly to experimental steps as well. Now, finally, what is the benefit of PrimePCR? Well, if we look at the assay performance and the system compatibility, we can see at the top left there that 90% of PrimePCR assays show 95 to 105% efficiency, exceptional consistency when compared to a home design primer, which is normally around 80 to 120% efficiency, reliable quantification without assay-specific optimization. And if you look at the dynamic range, which is at that bottom left, you can see a CQ of around 22, which is around a 4 million fold expression detection. This accurately measures both abundant and rare transcripts simultaneously, and is critical for heterogeneous samples with varying expression levels. And then if we look at compatibility, in the top right there, we can see a comparison between RT squared profile and TACMan. RT squared is QIAGEN and TACMan is Thermo Fisher. A consistent performance across 22 genes validates the reliability of our PrimePCR, and it uses the same biological conclusions regardless of the platform that you choose. And then in the bottom right, you can see under instrument compatibility, near perfect correlations, an R squared of 0.99 across 8 different thermal cyclers, works well on Bio-Rad systems and other manufacturers as well. And this can sort of help you doing multi-site studies with consistent results. And just to note that although all these real-time PCR instruments have different software, when you purchase a PrimePCR panel, there is software that you can make use of free of charge to sort of export your results from the real-time data of the system, and it will do the full sort of statistical analysis and ANOVA and all that through our PrimePCR analysis tool as well. Now, it's a lot of information, it's a lot of talk. What does it really mean in the real world? So I think we all know that real-time PCR is a powerful tool in life science research. However, the data it generates is only trusted if you've got sufficient attention to quality control and quality assurance, and you're making sure that at each step of the experimental workflow, you're monitoring these. Now, one particular example is highlighted in the slide, how wrong conclusions can be drawn from real-time PCR experiments that are not validated properly. So you can see here a study was published in which an association was claimed between the presence of the measles virus and gut pathology in children with developmental disorders. Ultimately, this data was then used to infer an association between autism and MMR vaccination, which we know is obviously very, very problematic. And this data was then multiple independent experts revealed sort of serious issues with this study, specifically what to do with real-time PCR data and the interpretation. And they refuted the claimed association. A consequence of this and other reports was that measles became endemic in the United Kingdom and some parts of the US going through a similar sort of process, mostly because of people refusing vaccination because of these specific claims. Now, the lack of documentation of a proper quality control in too many published real-time experiments can lead to a team of experts to develop sort of methods that they think is below par. So ultimately what happened was experts in the field came together to publish what is called the Markey guidelines. So that's the minimum information for publication of quantitative real-time PCR experiments guidelines. So amongst other things, these guidelines sort of describe the types of validation that need to be performed prior to the use of a real-time PCR assay. It includes things like in silico analysis, verifying the absence of an SNP, secondary structure confirmation, primer annealing regions. You need to make sure that you're doing wet lab validation to confirm PCR efficiency and specificity as well. And obviously these are now sort of a requirement whenever you publish real-time PCR work, which can be quite difficult to sort of work through if you're a researcher and a lot of additional effort required. If you compare this to the PrimePCR assays that we have from Bio-Rad, they are designed to meet these high quality standards. And these things include things like the Markey guidelines. And we do this by ensuring high quality validation and transparency throughout our experiments. Like I said, we wet lab validated every single one of our human, mouse, and rat assays. And the specifications we use require that every one of our samples has that high efficiency and a dynamic range of between 20 to 20 million copies. The assays have been temperature optimized and they all work at 60 degrees. So you can obviously run multiple assays on the same plate. They all work well together. They have been designed to be intron spanning where possible and to make sure that amplification is truly specific. Then we've also confirmed this with NGS to make sure that the sequence is there. So if we look at sort of the reporting that we give with every sort of PrimePCR assay, we can tell that we're really taking seriously setting a quality standard for real-time PCR assays by addressing the Markey's call for transparency and validation, by eliminating the need for end user sort of a validation, and ultimately saving our researchers time and resources, making sure that they are submitting high quality and confidence in their results. So that's the end of the presentation.
So thank you everyone for attending, and thank you very much, Luke, for a great presentation.
Thank you everyone. Thanks, Michelle.